All right, we'll go ahead and start. It's my great pleasure to introduce Lisa Stowers to present her work in this faculty lecture series. Lisa was recruited to Scripps in 2002 as part of our expansion into the areas of behavioral and sensory neurosciences at what is now the Doris Neurosciences Center. Lisa graduated from UC Davis, where she was named an outstanding senior. I dug real deep. She received her Ph.D. in cell and molecular biology from Harvard and remained at that venerable institution for postdoctoral studies with Catherine Dulac. There she showed using knockout technologies that TRPC, two ion channels in neurons of the innate olfactory vomeronasal organ are required to sense pheromones in mouse urine and to trigger sexually dimorphic behaviors in mice. And this paper was published in Science and was among the first to directly link molecular mechanisms to complex mammalian behaviors. I think Lisa's a quintessential TSRI recruit. Rather than pursuing low-hanging fruit, she invested her startup funds and took advantage of the physical and intellectual resources at Scripps to go after the ligands that were triggering these sensory neurons, because she knew that she needed these molecules in hand as essential tools for really identifying what are the neurons and for mapping the neuronal circuitry in this really simple behavioral circuit. So she took a hardcore biochemical approach and isolated these small peptides from male urine, and I can only imagine the process of collecting this stuff to do this purification. And so she also developed, this wasn't a simple biochemical assay, she had to develop a kind of a quantitative behavioral assay to assay column fractions to see where the activity was. So I think this was absolutely heroic effort. And the results of this 5-year study were published in Nature. As you'll see from the talk you'll hear today, she's continued this high-risk, high-impact approach and extended these studies to identify the molecules, neurons, and networks responsible for species and species-specific fear recognition and other behaviors. Lisa has been rewarded for these efforts. She was a Pew Scholar, a semifinalist in the last HHMI early career competition, and recently named an Ellisman Medical Foundation Senior Scholar in Neurosciences. As you'll see from the lecture, Lisa's an outstanding teacher. She's actively involved in the neurosciences program that's joint in the Mesa here, including a lot of lectures at UCSD. She's frequently invited to present her work at national and international meetings. I think you've been do more traveling than me internationally, Singapore and top institutions in the country, including Rockefeller, Cold Spring Harbor, Genelia Farms, and of course UT Southwestern. Lastly, Lisa is also an example of a true cell biologist. She knows that a mechanistic understanding of complex organismal behaviors or diseases begins with an understanding of the function of cells. And like all good cell biologists, she's an interdisciplinary scientist. She uses chemistry, biochemistry, molecular biology, genetics, imaging, quantitative analysis in diverse model systems. And so, good for you. And we look forward to her talk.
Okay, there's certain behaviors that are so conserved across evolution and stereotyped that just by looking at these still images, you may have an emotional empathy with these animals, and you may even be able to predict their next motor pattern. And these behaviors are not just evolutionarily conserved in their display, but also we know from ablation studies across many different species that the neural regions in the brain that generate these behaviors are also highly conserved. So we know that these behaviors are generated by neurons that are present in the amygdala and the hypothalamus, which is also known as the limbic system, or colloquially as the lizard brain. And furthermore, the appropriate initiation of these behaviors is essential for fitness. So too little fear is a bad thing. Too much fear is a bad thing. And the proper display of fear can save your life. And so these behaviors are incredibly fundamental for survival and fitness. They're conserved across evolution. We know where in the brain these neural circuits are acting to be able to generate these behaviors. But you may be surprised to learn that we don't know anything about the precise neurons that generate these behaviors. And the reason for that is that these regions of the brain are extremely heterogeneous. They're heterogeneous both in what they do. So the amygdala is known in humans to generate emotions, and in animals, it's thought to generate intensity across a valence of appetitive all the way down to aversive behavior. And the hypothalamus is known to mediate, contain what we call command neurons, or the neurons that are actually making a choice of which behavior to display amongst a variety of behaviors. You know, it's very hard to have effective reproductive behavior when you're incredibly afraid. And it's that opposition of these different types of survival behaviors that the hypothalamus is thought to make the decision as to the appropriate behavior for the individual environment. And so not only are these nuclei functionally diverse, but if we put an electrode into these nuclei and listen to the neural response during the behavior, or if we use a proxy for neural activity, which is an increase in the expression of an immediate early gene that increases its expression level after a neuron has been activated, so something like CFOS here. And that's represented in green in the cartoon and shown here in this image of a hypothalamus from a mouse that was undergoing fear defensive behavior. You can see that when we label for CFOS in these yellow dots here, it's very sparse and distributed, both in the amygdala and the hypothalamus. And what's probably a little bit difficult to see, but there's purple nuclei of the intermingled neurons amongst these yellow activated neurons. And we think that those neurons are encoding ensembles of neurons that are responsible for other aspects of behavior. And so the sparse distributed activity has been difficult to pin down and study. And moreover, if we do differential screens, we find that these nuclei contain neurons that are molecularly heterogeneous. And we and others haven't been able to identify any molecular markers that specifically correspond to these ensembles of neurons. And so because of that, we haven't been able to use traditional approaches that you would use in other sensory systems to be able to isolate and study these neurons of interest. What would be nice would be if we could capture these neurons using a genetic tool to be able to somehow label them. And Peter Cameron, a postdoc in my lab, in collaboration with the Maximov lab at the DNC, is trying to develop a new tool to be able to precisely mark and genetically label these neurons so that we could go in and functionally study them. But right now, all existing tools that others have tried to develop this approach also label behaviors that occur before and after the behavior of interest, and that confounds our interpretation of what's going on. So there isn't a precise tool right now. And in lieu of Peter's tool that he's developing, we've spent the last few years developing another approach that we think is not dependent on tool development. And that is, in the mouse, all of these behaviors happen to be initiated by olfaction. So the mouse is a special, unusual creature. Not every creature initiates these behaviors by olfaction, but they're nocturnal, and they rely on their sense of smell quite a bit. And if we take a wild-type animal and we put it in an environment that we know has an olfactory signature that is scary, we see, as I showed you in the past slide, this activation of immediate early genes that correlates with the display of the behavior. We and others have shown that there's a specialized region in the nose, a specialized suborgan that's present in all terrestrial vertebrates except for old world primates, of which humans are, so we don't have this, called the vomeral nasal organ. And if we knock out the sensory transduction channel in the vomeral, that's specific to the vomeral nasal organ, TRPC2, we don't see this corresponding increase in CFOS activity when the animal is placed in this scary environment, and we don't see them acting afraid. And so, furthermore, these neurons of the sensory neurons of the nose are very much like the immune cells in the immune system in that a T cell expresses a specific T cell receptor to be able to interact with a precise antigen. And in the olfactory system, olfactory sensory neurons each express one of a large subset, almost 1,500 G protein coupled receptors, and it's that unique expression of one of these receptors that enables the sensory neuron to precisely tell the brain which ligands it's detected in the environment. And so both the behavioral data and the promise of these specific receptors has led us to use an approach, a system where if we were able to use the ligands that generate a specific survival behavior to activate these sensory receptors and therefore identify the sensory receptors, we could use modern molecular and genetic tools to be able to mark not just the sensory neuron, but the whole circuit and genetically capture and study these neurons for this first time. So that's our overall approach, which, again, does not rely on technology development, so we're pretty sure we could do it. The only confounding problem is that when we began these studies, there were no known molecules of specific salience in the mouse. So these molecules had not been identified. And so for the past years, my lab has taken the approach that if we identify these molecules that have specialized meaning to the mouse, we could then use them to mark and study the neural circuits that are generating these really specialized innate behaviors. When we first started this, we were focusing our efforts on a single behavior, and in that case it was male-male aggression. But we quickly reasoned that that sort of line of research would allow us to become experts in one specific mouse behavior, but it wouldn't necessarily enable us to understand the general principles that generate information coding in the mouse. And so we're now taking a broad parallel comparative approach where we're studying 4 different behaviors in the lab. And the idea is to sort of take a forced approach where we're able to glean general principles of how the brain hardwires a response and generates innate behaviors and the common mechanisms across these behaviors, as well as a trees approach so that we can see the specific characteristics of the molecular mechanisms that make the generation of each one of these behaviors so special and unique. So today I'm going to tell you about our discovery of specific neurons that regulate 3 of these behaviors. Our 4th behavior we have a purified fraction for. We don't have the specific molecules, and I'm not going to discuss that further today. Okay, so our lab mice have a wonderful olfactory paradigm for innate fear. These mice have been isolated and inbred through Jackson Labs since the 1930s, so hundreds of generations, presumably without any exposure to any sort of other animal, or especially a potential predator. But we can take a cotton ball and rub it on the neck of the cat so that the cotton ball is odorized with the smell of the cat. And if we put it in the cage, the mouse will avoid the cotton ball. It will do risk assessment behavior, which means it gets up on its tippy toes and puts its tail straight back so that it's counterbalanced and it can run away quickly, and sort of dartingly sniffs the environment and runs away. So that's risk assessment. And it has a really rapid and robust increase in the stress hormone ACTH. So together, that repertoire is our defensive or innate fear behavior. Okay. So in the lab, the way that we can measure avoidance is to simply take this cotton ball, and if we just take a plain cotton ball or a cotton ball that's been odorized with a neutral odor like vanilla, and we put it in the cage, the mouse is just starved for activity and is so excited that something's new in his cage that he spends almost the whole assay, 20 of 30 minutes, directly in this area interacting with it, and very little time in the far end of the cage. But if we put in a cotton ball that's been odorized with this cat odor, this mouse, who's never experienced a cat before, will spend very little time with that exciting cotton ball and most of its time in the far corner. And concomitantly, if we measure his release of stress hormones, we find that in response to that cat odor, we get a very strong increase in ACTH that's identical to a very tight 30-minute restraint. So it's a very stressful environment, olfactory environment for this animal. So this is not unique to cat odor. We can show that it happens with rat odor, with snake odor. Others have shown similar types of behavior with weasel, ferret, owl. The mouse is afraid of odors from a lot of different potential carnivores. And we find that it happens here in blue is the response to cat odor in the wild type animals. On the left bars here for risk assessment behavior and avoidance behavior. Looks like it's backwards on these two. And you can see that in the wild type animals, we have the strong risk assessment and avoidance in response to all three odors, as well as the ACTH, which is not on this slide. But in these vomeral nasal defective animals that can't smell these odors, you can see that that response goes away to these odors. Okay. So I've told you that the response to these odors is innate. It requires some sort of chemical ligand, and the same response, the same generalized behavioral responses occur towards three different diverse species and requires the function of the VNO. But this is sort of an artificial paradigm that we've created. These poor mice, they're in their cage, strange things may happen. So is this really ethologically relevant? And in the wild, you know that a mouse is able to use all of his other senses to decide what's scary and what's not scary. And so we took a mouse and put it in a large open field arena with a dark black hiding box on one side, and it had an open area where the mouse was free to go in and out. It was safe in this dark black hiding box. And on the other side, we put in an anesthetized rat. And we asked, how does a wild type animal and a TRPC2 animal interact with this in this situation? And so these movies are a little strange because they're sped up really fast so that I can give you a sense for a long piece of the behavior and not just the specific piece. Okay, so these are filmed with an IR camera in the dark, so, but this is a completely black room. And you can see this wild type mouse. This is the anesthetized rat here. Here's its tail and its head. And the rat is breathing. He can see it. He could touch it if he wants to. It's a little bit stinky, like a rat would be, and its main nose is still functioning, so he can smell it. And he's spending most of his time in this corner. If I slowed it down to a regular speed, you would see that when he does come out, his behavior is very erratic and stressed. In contrast, when we take one of those TRPC2 mutants that it has its regular nose and all of its other senses, it just doesn't have this specialized nose. He spends a good deal of his time over investigating the rat. If I slowed it down, you would see that when he's investigating, he's very relaxed. His tail is relaxed. And in fact, of the animals that we tested in this paradigm, one became so relaxed over the 30 minutes, and the room was a little bit cold, that he actually cuddled up and went to sleep next to the rat, most likely to keep warm. And so when we've tested both the TRPC2 and the wild type animals for generalized anxiety, maybe this guy just doesn't have anxiety behavior. He has the normal levels of anxiety in an open field or in an elevated plus maze. So this guy is missing those cues to be able to tell him innately that he should be afraid of this guy. And without any other experience with the rat, he has no idea that the rat could do anything harmful to him. It's only those odor cues that give him the innate ability to detect that. And so that is, the data is quantified here. Okay, so if we take the odor cues and we look at, again, the immediate early gene proxy for neural activity in the brain, normally, in response to a control odor, there's very little background activity, and if we expose to cat, rat, or snake odor, we get this bright, speckled CFOS activity pattern that is not present in the mutant animals, the brains of the mutant animals. And so we really think that the problem with the mutant animals is that they're not getting the sensory information to be able to lead into these survival circuits. And so to be able to identify these cues, we started with rat urine, which potently initiates these behaviors, and it's something that we could easily fractionate. And we first fractionated it into just different size components, and we found that all of the avoidance and risk assessment behavior fractionated with the high molecular weight activity, and that the high molecular weight was able to initiate a robust activation of that first relay in the brain. There is some, especially, it's hard to see from the back, I'm sure, but there is some CFOS activity in response to the low molecular weight, And we know that there's other stimuli in the low molecular weight, but that's not of interest to us. We're really only interested in the activity that has the bioactivity associated with it. So we took that high molecular weight and just did a mass spec on it. We had one prominent peak, and we could fractionate that peak by FPLC and be able to compare the bioactivity of this peak to the other fractions that we got by FPLC. And this mass was known in, we could match it up to the rat genome. And we found that it was something called a rat MUP, which is excreted, it's known to be excreted into rat urine. And so we went ahead and just made, it's a protein, and we went ahead and made a recombinant version of this protein in bacteria and tested that as well. And if we test the recombinant version or that FPLC peak, if we perfuse it across the primary sensory neurons of the vomoral nasal organ, we can show that indeed they activate the neurons as shown by this heat map of a single neuron here as it's responding, or this graphical representation. Each of these lines is a single neuron, and you can see that the neurons that respond to the native high molecular weight fraction are the same neurons that are responding to this purified peak and the bacterially produced recombinant protein. And moreover, when we test both the purified peak as well as the bacterially produced protein, we find that that protein does account for the majority of the bioactivity that was originally in the rat urine, as well as it generates that stress response just when we just have this bacterially produced protein on a cotton ball. So we're very pleased that this is a signal that promotes this fear behavior, and that to the mouse, it's this protein that represents the presence of this animal animal in its environment and generates this innate fear. But I mentioned one of the interesting things about this behavior was that there's multiple animals that our mice will each innately respond to. And it was postulated that perhaps the mouse is responding to a metabolite of carnivore metabolism, but these MUPs are really not a byproduct of metabolism at all. So we're very curious to understand what is the salient ligand in another animal so that we can compare how the ligands are evolved to initiate these responses. The first swabs we didn't start our fractionation procedure with because they were just very difficult to be able to analyze the material on them. But one day, as we were swabbing the cat bit the cotton ball, and my graduate student was insightful enough to note that in her notebook. And she noticed, as we did the assays, that this cotton ball gave amazing behavioral activity and really strongly activated CFOS in the brain. And so there had been speculation in the literature. We had always done neck swabs because speculation in the literature that back swabs were less effective, and perhaps it was because the cat grooms himself very predominantly in the neck region, and that perhaps the salient cues were actually in saliva. And so being Southern California, we were able to go to a cat dentist and obtain some cat saliva. And we also just did a quick literature search on saliva and found that saliva actually contains the rat ortholog of this protein. So it's not emitted in the urine of rats, it's actually emitted in the saliva of rats. So we made a bacterially produced recombinant version of that. And as you can see, both the cat saliva in the second column here and the recombinant version of that protein were sufficient to elicit the avoidance and the risk assessment, as well as the ACTH induction. It activates the brain, and importantly, it activates it in a TRPC2 dependent manner. And so overall, we think we found the beginning of a code that allows the mouse to be able to detect other species and initiate innate fear. And we've identified this ligand that we were looking for. And we can find a specific ligand that we know through the biology of the olfactory receptors will activate a specific olfactory neural circuit and will generate the defensive behavior. Okay, so I'm going to switch gears abruptly here and tell you about another story where we did a similar approach to identify ligands. And those are the ligands that generate territorial defense. And so males across many terrestrial vertebrate species display this behavior. They use it to be able to establish their territory, to attract mates, or to fight for resources. It's usually gender dimorphic. Females don't do this behavior, and it's usually testosterone dependent. And mice do this behavior as well. And so we can do this in the lab by simply placing a mouse in a cage by himself for about a week, and he starts to think of that cage as his territory. So he's the resident. And if we put another male into his cage, the resident will fight him. As you would guess, if you put a female into the cage, the resident will not fight her. And if we put a juvenile into the cage, the resident will not fight him. And the difference between the adult male and the juvenile male is the ability to produce testosterone, and that allows him to produce the signals that cause the fighting. And so if we castrate an adult male and then put him in the cage, he doesn't provoke aggression. But if we castrate him and then paint a little bit of male urine onto his back, he now is carrying around the cues that says that he's an intact male, and the resident will fight him. And this guy's a really good proxy because castrated males not only don't emit the aggression-promoting pheromones, but they also don't respond to them. And so this guy doesn't initiate any behavior, and he, even when attacked, he will just run. He won't fight back. And so any aggression that we see during the assay, we know is coming from the resident. And we can use this castrated male as a platform, like we did the cotton ball, to be able to identify the salient cues. And so this is the behavior. This is a wild-type male in with a castrated male. Let's see, this is the wild-type male. This is the castrated animal. This is 5 minutes into a 10-minute assay. And this male is just very calm. You can see he doesn't really care that another guy's in his cage. He's interested that his feeding tray's out, and he's looking around. His tail's relaxed, and he's just kind of relaxed. In contrast, if we take the same two animals about a half hour later, and this time we paint a little bit of urine from an intact male onto the same guy's back, and we put them together, this is the behavior. So this is, again, 5 minutes into the 10-minute assay. It's very easy to score. It's very robust behavior. It's really aggressive. These male mice are actually quite strong, and they have very sharp teeth. And if you look really closely, or if we were to slow it down, you can see that the castrated animal isn't initiating any of this aggression. So those are the behaviors that we're looking at. And we did the same tricks that I just showed you with the fear behavior, where we did biochemical fractionation with behavioral analysis and neuronal activation, and we were able to identify the ligands. And so the ligands turn out to be the orthologs of those proteins that I just described, the major urinary proteins. And in the mouse, they're produced in the male liver, secreted into the bloodstream, and excreted into the urine. And they're predominantly regulated by thyroxine, growth hormone, and testosterone. And so females produce very little of these, as would be expected by this type of cue. So it turns out, in the cat, there's only one ortholog of these major urinary proteins. But in the mouse, the cluster has recently expanded, and there are 21 variants of this proteins. And there's a central region to the genomic cluster that's duplicated quite recently. And when we plot the differences in predicted amino acid residues onto the solved structure of these in magenta, you can see that across all of these 15 different MUPs, there are only these 7 differences. And most interestingly, these 7 differences don't all cluster in a region that would look like a receptor binding domain. They're sort of spread out across this face of this protein. And then there's these more evolutionarily ancient MUPs that are more divergent. And so even though there's a lot of these MUPs, we were very curious, what's the coding logic of them? Are they each able to carry redundant information? It looks like perhaps most of them are just redundant and not capable of providing any specific code. So to be able to look at this, we simply took the calcium imaging assay that I showed you previously, and to individual neurons shown here by these traces, we perfused recombinant versions of these individual mouse MUPs over the neurons, and then just a control of the pool of the recombinant versions. So these 4 neurons respond to the pool, but they're responding to different variants in the pool. So this first one is responding to MUP3, but not MUP8, whereas this one's responding to MUP8, but not 17 or 3, et cetera. So even though these, and these 3 in magenta correspond to this cluster where they're only 1 or 2 amino acids different from each other, compared to this one here that is significantly different. So there's the potential for these ligands to actually generate a different neural response or carry different information when the mouse detects them. And so we asked, what's generating the aggressive behavior? And when we look at just a fraction here, and we've since looked at more of the individual MUPs, the black bar is the amount of aggression that we see in response in a behavioral assay. And the second bar is a negative control. And all of the individual MUPs pretty much give us a behavior that's very similar to the negative control, except for MUP25 here, which recapitulates all the behavioral activity that we see in the pooled MUPs. And so that MUP is a special MUP, and it's carrying special information in comparison to all the other MUPs that are in the urine. So one of the unusual things about these MUPs is that even though there's 21 of them, males don't express all 21 of them. Each male expresses anywhere between 4 and 12 of them. And if you look at wild-caught brothers on an isoelectric focusing gel here, where you're able to separate by charge, you can see that the individual MUPs from wild-caught brothers here, the MUP variants vary from individual to individual. And in the lab, because of our inbreeding, the MUPs have become stabilized to a specific strain. And so all of the C57 black mice that we use express this combination of 5 MUPs here, denoted by these stars that we've put here. Whereas if we look at a different strain, BALB-C, it's expressing a different combination of MUPs. And remarkably, that one that we showed generated the aggressive behavior is present in the strain that we normally use, C57 black, but in this other strain, we can't find a trace of it. And so this provides us a natural loss of function mutant, a mouse that's never experienced this MUP before. And we can ask, does MUP25 have innate salience to an animal? Or is this animal learning through development and experience with this MUP that it should act aggressively towards it? And so we simply did the behavioral analysis with that other strain, the BALB-C, that doesn't express the MUP. And while the behavior isn't as robust as total urine, we definitely see an increase in aggressive activity compared to when we do the same behavioral analysis with the other MUPs that don't generate the behavior in our strain that emits it. And so we believe that there is some salient information inherent to this molecule, and that MUP is specific to generate aggression. So now we have 2 cues, and I've told you, and they're acting very similarly. We find a single cue, we think that it's activating a specific population of neurons, and it's leading to one or another behaviors. But there's more questions that we can ask about how behavior is encoded with the MUPs. And that is, I told you that individual males emit different MUP proteins. And so this would be odd if some guys get the aggression-promoting MUPs and other guys don't. And we don't know what the other ones are doing, but what if some are things that are really important and some guys get them and other guys don't? And what is the logic of this? This seems like a weird way to establish a logical representation of a social behavior network. And so it had been previously speculated that there should be some olfactory mechanism emitted and detected between mice that generate individual recognition. Now, in mice need to know the difference between mouse A and mouse B, or Bob and Harry, to be able to control their social behavior and their response towards females. And that is, there's a very strong dominant-subordinate hierarchy among mice. And any individual male has to know whether he's dominant to male X or subordinate to him. If he's dominant to him, then he's going to have very aggressive behavior. And if he's subordinate, he's going to make himself scarce. And one of the ways we can visualize this is that is by the pattern of their urine markings in their environment. So it was previously observed back in the 70s that if you put two males together, so that one becomes dominant and one becomes subordinate, and then you visualize their pattern of urine marking, and here it's visualized by UV illumination, you can see that the dominant guy advertises himself quite profoundly, whereas the subordinate guy basically hides in the corner and has this characteristic display of urine marking. So the reason that the dominant guy does this is apparently the urine is very attractive to female. And so if you're dominant, you want to display the fact that you're there and you're in charge. And so males display mark predominantly to female urine. And if another male, if male A marks in this way, and male B comes along and male B is actually dominant to him, he will countermark, or he will place his urine marks on top of it. He will be the freshest, and the female will then find him. Will not, a subordinate will never countermark a dominant. Okay. All right. Okay, so the male has to know who he is and who the other guys are to make this work, and that's a test for individuality. Okay. So we wanted to see if we could, if these MUPs were directly regulating this individuality. And instead of placing the animals, so in this original assay, the animals were in the same arena with a wire mesh, so they couldn't interact with each other. But they could hear and smell each other in general. And we wanted to simplify this so that we weren't activating other sorts of behavioral responses. And so we just took a piece of Whatman paper and put a spot of urine down in the middle. And then we allow the animal into a cage where this Whatman paper is on the bottom of the cage for 10 minutes. And then we develop the urine with nihydrin and scan it in and quantify the amount of color that we see. And if we put a drop of urine compared to a drop of water, you can see that the dominant male marks in this characteristic pattern, just like was shown previously, and that subordinate males do not mark to the urine. Both males mark to female urine, as would be expected if they're trying to advertise, and the subordinate does it, so he's capable of doing it. He does it when he doesn't think that another male is around. Okay, so that's our assay. And so again, we fractionated just to low and high, and the MUP-containing fraction does generate countermarking behavior. And so if we do a more clean test with the recombinant MUPs recapitulate the entire urine activity. Okay, so the question is, are these MUPs acting in the same manner as the aggression-promoting MUPs? That is there a specific MUP that says you should countermark, or is there another code? One of the confounding things is that self-urine doesn't promote countermarking, and so in our self-urine strain, is there a countermarking MUP, and how do you encode the individuality? And so we tested the recombinant MUPs that are the profile of the self strain that we use, and they do not generate the countermarkings consistent with what we know of the biology. And so generally what I've shown you is that if you emit one combination of MUPs and you detect a non-self combination of them, you get countermarking, but if you detect the same combination of them, you don't get countermarking. If we take the MUPs and test them individually, as we did for the aggression assay, we find that no single MUP is special. They're all able to generate countermarking at a level of total urine or at the level of the pool of the MUPs. And so this is very different than what we saw before with the aggression ligand system. And so if you, again, take one of these single MUPs and you detect it, you can generate the countermarking. Now, we were thinking that perhaps you didn't countermark to your own MUPs because there's sensory habituation. Like if you walk into a room of baked bread and it first smells wonderful, and after 10 minutes or so, you really can't detect it very much because your sensory system has habituated and is no longer responding to that stimuli. And we thought that perhaps the mouse was doing the same thing. He's producing these MUPs all the time. They're in his environment. And so perhaps he's simply habituating to his own MUPs. And that would be a very nice code. So in our repertoire of MUPs that are emitted by our self and non-self strains, so this is the self strain that we're using for this assay, you can see that there's some overlap. Just like in these wild-caught brothers, there's some MUPs that are common amongst individuals. And in this case, this MUP here, which corresponds to what we call MUP3, happens to be expressed in both the self and non-self strains. And so if it was simple habituation, we would expect that if we present the mouse with just this MUP, MUP3, that it wouldn't countermark to it. But in fact, he does, just like he does to all the other individual non-self MUPs. Okay, so now we have this, and that if you take one of these guys out, he countermarks to this. And what we think is happening is that the mouse is actually making, it is very much like a barcode, and you need all the pieces of information to be able to have significance and for the mouse to be able to detect self from not self and A from B. And so we believe that the mouse is representing this combination versus this combination to be able to have behavioral significance. And so we're currently doing subtractive comparisons as well as add-back comparisons to try and understand whether this is possibly the case. And from there, we think this is a very exciting behavioral paradigm to study, because something like MUP3, which doesn't elicit the behavior here but does elicit the behavior here, if we were to study the circuit activated by MUP3, we would then be able to study basically the individuality response, and we're very excited by that. And so this is a very interesting behavior because the male has to make several choices. First, he has to decide whether the odor cues are males and females, and if it's female, he's going to countermark. If it's male, he has to decide whether it's self or not self, and if it's self, he doesn't countermark. And if it is non-self, he has to decide whether he's dominant or subordinate to this odor, and if he's dominant to it, then he will countermark. And so these sort of neural decisions we find very interesting. And overall, however, we think that this ligand, the detection of these ligands is acting on a different set of rules than what we identified for both aggression and fear. And that is that the animal has to be able to measure and evaluate a group of neurons, a group of ligands simultaneously. So in the last few minutes, I'm going to change gears once again and tell you about what is arguably the most important behavior in mammals, and that is the ability to suckle. So every individual has to suckle immediately upon birth. And babies come out knowing how to do this, and it's just remarkable. So in almost every organism, there has been hints that there's an olfactory component to it. And about 4 years ago, a group identified a single small compound that's released from the mammary gland of the rabbit and fits all the criteria for a pheromone. You present it to the mice on to the rabbits on a glass rod, and they immediately start suckling the glass rod. So it's very nice single molecule, but the rabbit isn't a very good model organism for us to understand the corresponding neural response. And so we wanted to be able to find such a pheromone in the mouse so that we could use the molecular genetic tools in the mouse to be able to study this more appropriately. And so this is the way that we do the behavior. It's very low tech. We have a mom, and she's anesthetized, and her ventrum's shaved here. And we have a pup, and he searches, and we basically time from when we place the pup at the beginning of this video, how long it takes him to find the nipple and start these characteristic jaw movements that are characteristic of suckling. Okay, so as you can see, this thumb is getting closer and closer, and it looks like it's pushing him into the nipple. And I'm going to try and convince you that that's not the case. You can't make a pup suckle, but he's merely stabilizing these pups because they're so small they can't reach the nipple. Okay, so the way that I've quantified all this data is that each horizontal tick here is an individual animal. This is a 90-second assay, and you can see a wild-type animal takes about, on average, 20 seconds to start to suckle. If we take those mutants that I showed you before that don't do fear behavior and they don't do the aggression behavior because they don't have a functioning vomeral nasal system, they suckle just fine. But if we take mutants that do have an intact vomeral nasal system but don't have an intact main olfactory epithelium, which is the system that we use to smell things like pizza and banana. These guys don't suckle. And this has been known for a long time. These mice are notoriously difficult to work with in the lab, because if you leave these mice just by themselves, unattended in the cage in a natural setting with the mom, they all die without milk in their belly. They just don't suckle. So this amount of suckling, which is do not suckle in 90 seconds, and there's a latency of these few guys that do suckle that's off the 20 seconds. It looks fairly favorable here, but in the natural conditions, these guys are all dead. So it's pretty bad. Now, if we take the mom and we simply wash her ventrum with a little bit of water, and now we've extended the assay for 15 minutes, you can see that on a washed nipple, most of the animals now do not suckle. And if, I'm sorry I didn't put it up here, but if we compare this to the CNGA2 mice, it completely copies the CNGA2 mice. And so we know that this now provides a really good platform for us to isolate the olfactory cues by just adding them back to the nipple. So all of the rest of our assays are in that shorter 90-second format. The pups, if we go 15 minutes per pup, they all dehydrate. It's just not effective use of animals. And we did a lot of careful statistical analysis to be sure that this behavior can be recapitulated in the 90 seconds. By cutting it off at 90 seconds, our statistics aren't as strong. So, okay, this is what I showed you before. Unwashed nipple, the pups suckle rapidly. Washed nipple, they do not suckle. If we add back to this washed nipple several native odor sources. So when a mouse is born, the mom rips open the amniotic sac and vigorously licks the pup to be able to expel the amniotic fluid so that the pup begins to breathe. And then she cleans her own ventrum and then crouches over the pups to nurse them. And so during the course of that, she exposes the pup is exposed to amniotic fluid, maternal saliva, and eventually milk. And all these have been expected to be sources of pheromones in the literature. And you can see if we add back each one of these onto the nipple, then the pup rapidly suckles in response to any of these. But it's not just anything that we add back, because if we add back maternal urine, or interestingly, virgin saliva, or a whole panel of non-relevant odors, they don't suckle to just anything novel. Okay. So we had reason to believe that the pups that we had used in the assays that I've shown you up to now were all 12 hours old. They had all suckled before and had milk in their belly. And we thought perhaps the experience was conditioning learning to non-relevant odors. The act of suckling is incredibly reinforcing, both with the sweetness of the milk and the tactile response and the heat of the mom. And so we were really interested in just the innate cues that causes the first suckling. And so to analyze this, we did a C-section on the pups about a half day before they were supposed to be born. And we brushed the pups with a paintbrush that just had water on it to be able to mimic that licking of the mom and be able to initiate that first respiration. And then we swabbed the washed nipple with the various odor sources. And now when we do this, this is the washed nipple, and this is the washed nipple plus the maternal saliva, which previously generated suckling activity, milk, and here's colostrum. We find that none of these now are sufficient to generate innate suckling. Amniotic fluid does, which is good, which means that our pups are healthy and that they're able to suckle in this assay. And if we instead do a C-section on pups, and instead of brushing them just with water to stimulate them, we give them artificial odor stimulation with singularly, one pup would get saliva, say, and then we would challenge him with saliva on the washed nipple. And when we do that, okay, let's see, this is the washed nipple. This is the data that I showed you before. And if we add back now and artificially add maternal saliva, he now suckles in response to it. Milk or colostrum, they're now suckling in response to it. So this clarified the confusion as to how these multiple sources would generate the behavior. And it also simplified our task. We weren't looking for multiple things or a common thing. We simply had to look at amniotic fluid to find the cue. Okay, so in this case, the first thing we did was just lyophilize amniotic fluid. And when we lyophilize it, the bioactivity goes away. And that tells us that the ligands are volatile. They're not MUPs. This time we did a 3-way fractionation into 3 different molecular weight sizes. And when we paint them back onto the wash nipple, neither of the three fractions are each sufficient to generate the behavior. And it's only when we add a combination of the two lower molecular weight fractions together that we're able to generate the bioactivity. So that tells us that it's a blend of at least two different ligands. Now, this was the limit of our capabilities to be able to size fractionate. These experiments are very difficult to be able to sequentially take a very logical molecular fractionation approach as we're doing with the estrus pheromone, which is also a low molecular weight substance. And so the way that they identified the rabbit pheromone in this case and several other pheromones subsequently has been to couple the mouse to the port of a mouse, port of a mass spec, and to be able to analyze activity, behavioral activity, as each individual peak is coming off the spec. Now, this is really difficult, especially with a mouse pup who can't live very long without suckling, and we don't want to give him experience. And when you imagine doing it with at least two different things, you're now running offset peaks against each other, trying to find the two that would have behavioral activity. And from this, we don't know that it's necessarily two. It could be 6 different things. And so from a technology point of view, we weren't able to find anybody that would collaborate with us to help us do this. And we took another approach, and by doing several other assays on both the neurons of the animals and through the behavior, we began to question whether there was a traditional pheromone at play here in the mouse. And so a traditional pheromone is defined as a particular molecule that is expressed throughout all members of the same species and generates some predetermined activity. And so the MUPs are a classic example of this. So in the literature, there's another kind of odor, and that's called a signature odor. And that's a chemical mixture that varies between individuals, and animals have to interact with each other and learn the nature of the signature odor for it to have any sort of significance. But under these certain contexts, the signature odor generates a behavior that's very much like an innate behavior. And so the only example of a signature odor in the literature is that in sheep. And so sheep are born pretty much around the same time. A whole flock is going to birth around the same time, and sheep are mobile as soon as they're born. And so they're born, they run away from their mom, and then they have to find the mom again. They don't care which mom they find to nurse from, but the mom only wants to nurse her sheep. It's a bad use of resources to nurse somebody else's lamb. And so if a strange lamb comes up to her, she'll kick it away. It's not very nice. But she will recognize her lamb based on odor interactions. And the odors have never been characterized, nor has the neural response, but it's this learning paradigm through behavioral manipulations that makes one speculate that there's something called this signature odor. Okay, now this is useful to us because though we can't do the sequential fractionation of the amniotic fluid, and we couldn't figure out a way to raise the pups without amniotic fluid, we reasoned that if we were looking for a pheromone, particular odor molecules, that the activity of a pheromone is not expected to be diminished if we alter the surrounding and olfactory environment, if we changed some aspect of the rest of the odor cues. However, a signature odor, if it's a signature odor, it's by definition expected to be altered if you add in any non-relevant cue to it. Okay, so we could use this to just tell us the type of molecule we're dealing with, and if we have to go back and think of a new way to get a pheromone, at least we know what we're dealing with. So we were able to alter the amniotic fluid profile just by altering the diets of the moms. So we fed moms either water or water that had been odorized with vanilla. And in collaboration with Bill Webb here at Scripps, we were able to do XCMS. And here's an example of a differential metabolite. And you may or may not be able to see a haze of gray balls along this axis here. And these are things, metabolites that are found to be in common between the vanilla odorized water fed moms and the regular water fed moms. So all of the amniotic fluid is the same. There's about 3,000 metabolites. There are these small differences on one axis or the other axis shown in red and black here. And they're quantified here, and as you can see, overall, we've changed less than 1% of the metabolites. And importantly, if we take these amniotic fluids and we use them to perfuse the primary MOE neurons of these newborn animals, we find that most of these fluids activate the same neurons, but there's a small subset of neurons that's activated differentially by the vanilla odorized amniotic fluid compared to the regular amniotic fluid. And so that tells us that it's not just our metabolite composition that we've altered, but actually the odor profile that we've altered. We've changed the odorants, but just slightly. Okay, so here's the crazy experiment. The idea is that we feed moms either water that we've odorized with garlic, or water that we've odorized with vanillin. Then, partway through gestation, we harvest some of the amniotic fluid, and we do a C-section on the mice just as we've been doing in the previous assays. And we expose these mice that have been gestated in one environment to all nipples. The individual mice just get one assay, but they're exposed to either the regular amniotic fluid, garlic odorized amniotic fluid, or vanillin odorized amniotic fluid. And the hypothesis is that if there indeed was a pheromone, and we should just try harder with our fractionation, that we would see activity under all circumstances. But if there was a signature odor, perhaps it would bias the behavior towards the specific environment in which it had the experience. And indeed, that's what we saw. So the data, this is sort of complex, but what you should focus on here is the pups from the moms that were fed vanillin specifically suckle in response to the vanillin amniotic fluid. The pups from the moms that were fed garlic specifically suckle to the garlic amniotic fluid. And I think most importantly, the pups from the mom that were fed the water, maybe these guys had some developmental defect, but these guys developed just normally. They specifically suckled to the amniotic fluid from which they were gestated in. It didn't have that small change in the neutral composition of odors. And so we think this is really interesting. Even though we did not find the salient ligand that we're looking for, we don't think that there is one. And we actually think in the case of mice that this is a beautiful explanation of biology. Because what we were seeing through the VNO is that those pheromones that went through the VNO are recognized by receptors that have very strong ligand feature extraction. They only respond to the correct stimuli. And this requires both molecular hardwiring of the response and developmental hardwiring, and the animal has to be in the right state to be able to respond to the pheromone. And this is great because it maximizes the discrimination, the regulation of the behavior. You don't want an animal to be afraid when it shouldn't or to be fighting when it shouldn't. Okay. But with this behavior, you just want it to happen. You want it to happen, and it doesn't just happen by itself. It needs specific cues. And so we think that this is going through this other system, the main olfactory system, whose job throughout the rest of the animal's life is associative learning, and that it's utilizing, we think, signature odors that requires this olfactory conditioning and learning sometime throughout prior to the first suckling. And that this is great because it, in theory, would have both a molecular and developmental plasticity, and that is, if the mom doesn't produce the right cue, it'll still happen. If the pup is underdeveloped and doesn't have the capability to respond precisely, it'll still happen. It just has to do a sensory pattern matching to be able to get it right and initiate the behavior. And we think that this perhaps would maximize the probability of initiating this first behavior. And so overall, we now have another way to generate what appears to be innate behavior. And that is through this learning repertoire and pattern matching of ligands that don't necessarily have salience to every individual of the species. And so while we're still poised right now to be able to take these ligands, especially those for fear, aggression, and throughout the countermarking, and to be able to genetically manipulate their precise receptors. We can use the ligands to precisely activate the behaviors in absence of activating obscuring neural activity throughout the brain. And we can use them not only just to generate the behavior, but to mark and study these circuits. And we're really excited to be able to do that in the future. And so The work on the fear behavior was done by these two postdocs in my lab. Fabio Pappas is now back in his home country of Brazil as a professor at the University of Campinas. And in addition, Darren also did the entire suckling story in collaboration with John Nye and Lisa Brunet at UC Berkeley. And Darren now has his own lab at the Sanger Center in Cambridge. And the countermarking story has been worked on exclusively by Angel Deep Carr, who's a TSRI graduate student in my lab. So thank you.